So this is Kiel. This is where I'm based. It's by the northern part of Germany, facing the Baltic Sea. And when you plan to come to Germany, you should come there in summer. Then it's beautiful. In winter, it's somehow debatable. But this is just 10 minutes from the lab. So this is really a great place. Okay. So the lecture of today is interleukin-6 in health and disease. And you probably realize that only few cytokines are really very, very important in autoinflammatory diseases. And one of these cytokines is interleukin-6, besides, let's say, TNF-alpha, and perhaps also besides interleukin-1. But IL-6, we think, is a very, very important cytokine, and I will tell you something about the biology of IL-6, which you probably don't know and which is very complex, I admit, but it's important to understand this. And you will also see that this cytokine is really involved in many activities in the body, and when you block IL-6, and that might also be true for other cytokines, when you block IL-6, you should really be familiar with the biology of this cytokine. Okay, so my disclosure, so this is also listed in the program. I don't have to go through this in detail, I guess. And we start with the cytokine interleukin-6. This is the protein. This is a three-dimensional structure which has been solved. It consists of 4 helices with a particular fold, 2 go up and 2 go down. And this is actually, for a biochemist, you should know I'm a biochemist, not a physician. For a biochemist, this is one of the very few examples that a protein fold has been reserved for function of a protein. So all proteins which have this kind of fold are mediators or cytokines or hormones or communication molecules. So obviously you know that IL-6 is involved in many, perhaps even all, chronic inflammatory states, that the cytokine is important for the function of the innate and adaptive immune system, that it's a target for anti-inflammatory biologics. You're probably familiar with the antibody tocilizumab, which targets the human IL-6 receptor. That there's a reason for this. If you want to discuss it in the end, I can explain it to you. And this now has been approved in more than 100 countries for the treatment of rheumatoid arthritis and other inflammatory diseases, underlining the importance of this cytokine for autoimmunity. Now, there are a couple of things which you probably are not that familiar with, and I will try to explain these to you. So this is IL-6. This is the so-called IL-6 receptor, and IL-6 actually binds to this receptor on the cell surface. This would be a cellular membrane, and here you would, in the cell, then have signal transduction. Now, whenever IL-6 binds to the IL-6 receptor, then this complex associates with a second protein which is called GP130. And now it's very important that you remember 2 facts. One is IL-6 has only a measurable affinity for this protein, for the receptor, but not at all for GP130. This is very important. IL-6 only binds to the receptor, not to GP130. And second point is GP130 is on all of your cells, whereas the IL-6 receptor is only on very, very few cells, namely on hepatocytes and some very few leukocytes. This you should remember. And if that is true, then you can actually divide the world into 2 sorts of cells. So one cell would be IL-6 responsive because it has this IL-6 receptor and GP130, here. And the second part is not responsive because it only has GP130 but no receptor, cannot respond to the cytokine. This sounds very easy, very clear-cut, but there is something wrong with it because you know probably that during inflammatory states, endothelial cells and smooth muscle cells are very strongly responding to IL-6. So there is something missing. We were actually looking for this. And what is missing actually is, so here again you have this what we now call the classic pathway, IL6 receptor, GP130. Here you have the signaling which takes place within the cell. You have heard about this already. But then we noted there's a second reaction which has been overlooked, and that is the receptor. This one is actually the same as this one. This receptor is actually cleaved by a metalloprotease which is called ADAM17. And when it's cleaved, then you generate a soluble receptor because you just cleave it off here by the membrane, and now it's a soluble protein. And the soluble protein can actually still bind the ligand IL6. That is known for such soluble receptors. We know that many proteins are actually cleaved from the cell surface, but now comes something which is unique to this complex here, and that is this complex can actually bind on a cell which has only GP130 and which would not be responsive to the cytokine. So this binds now on such a cell, and it not only binds, it induces signaling. So that means that this cell here, by cleaving this receptor, makes a different cell or a different tissue responsive to the cytokine. And this principle we found kind of attractive or fascinating, so we called this process, we called this process trans signaling because it goes in trance. This cell makes the soluble receptor, and together with IL6, a different cell is activated. And this is true, this mechanism we think, but I will show you a little bit more, for endothelial cells, smooth muscle cells, and some epithelial cells. And obviously, this kind of switch seems to be performed by this protease, ADAM17, which thereby switches from this classic to trans signaling. Therefore, we think this protease is also very, very important and should be studied and might even offer a therapeutic target. Okay, now, when you want to make a point in biochemistry or molecular medicine, you need tools to really pin it down. And so we set out to generate a couple of tools to make the point that this trans signaling is not only a beautiful petri dish artifact, but that it really occurs in vivo. So one of the tools we made, I have no time to go through all of these, but this is important. So what we made is a fusion protein of the entire extracellular portion of GP130 fused to the constant portion of a human antibody, a so-called FC. And when you do this, then you can, as some of you might know, you can precipitate this protein with protein A saccharose because protein A binds to this constant portion of an antibody. So we could make the following experiment. We made ourselves a radio-labeled soluble IL6 receptor, human soluble IL6 receptor. Now we precipitate this protein with protein A saccharose, and we do this in the presence or absence of IL6. And very clear cut, when you have no IL6 in there, you do not precipitate a single count, just nothing. But whenever you put IL6, GP130, and soluble receptor together, and then no matter what species you mix, you always can precipitate a complex. This means IL6, as I told you, does not directly interact with GP130. It needs this protein, the soluble IL6 receptor. And this has very important consequences, which are shown here. When you try out cells which now have this composition, which you know, IL6 receptor, GP130, and this cell, obviously, you have to stimulate with IL6, and now the cell proliferates, which you can measure very nicely with incorporation of thymidine into DNA. And when you do this in the presence of this soluble GP130 protein, you see that the proliferation of the cells is not affected at all. No matter how much GP130 you put into the petri dish, the cells just keep on proliferating. But when you do the very same experiment with such a cell which you also know already, a cell which only expresses GP130, And now, obviously, from what I told you, this cell has to be stimulated with IL-6 and soluble receptor. You do this, and you do this again in the presence of this soluble GP130 Fc protein. And now, in contrast to what you see here, very tiny concentrations of this soluble GP130 completely block the proliferation, kind of indicating that this protein here is able to discriminate between this situation, stimulating via a membrane-bound receptor, and this stimulation via a soluble receptor. And that means now we have a tool to sort of say this kind of activity relies on this or on that. Okay. And obviously, we also studied, but I have no time to go into this, we studied very much the activity of this protease ADAM17, which we think really orchestrates many signaling pathways, including the one of IL6, TNF-alpha, and also EGF receptor. You might know EGF receptor is stimulated by ligands which are all membrane-bound, and they need to be cleaved by a protease, and this protease, again, is ADAM17, and TNF-alpha, you also know, is a transmembrane protein, and whenever it needs to be activated or be systemically active, it needs to be cleaved, and again, this is ADAM17. So we made mice which are hypomorphic for ADAM17, and we found many things, but as I said, I have no time to go into this. Now comes one more thing, which is very important for the understanding of the biology of IL-6, and this is what we call the buffer in the blood. So IL-6 binds to the IL-6 receptor with a certain affinity, which is 10 to the minus 9 molar or 1 nanomolar, which is a medium-high affinity. There are ligands which even bind better to their receptors. There are many ligands which bind less good. But, okay, that we should reason. This is known, that can be measured, 1 nanomolar. But now, when you measure how good IL-6 and the receptor complex bind to GP130, and no matter whether it's membrane-bound or soluble, then you see that this is 10 picomolar. So this is the affinity of this to this is 100 times higher than the affinity from this to this. Meaning, when all three components are present, then IL-6 will immediately bind to this, and then this will bind to this, and as we just saw, this then will, when it's soluble, be neutralized. So that is something which might be important. But also important is actually what are the normal concentration you find these proteins in the human blood. So IL-6 is very, very low. It's 1 to 5 picogram per mil. This is barely detectable. It's just about the limit of detection. But curiously, this protein, the soluble receptor, is found at 40 to 75 nanograms per mil, which is about 10,000 times higher than this one. And curiously again, a soluble form of GP130, a natural soluble form, is found at 250 to 400 nanograms, again, almost a factor of 10 higher. And we think that these two have to do with the in vivo biologic activity of the cytokine. We call it actually the buffer of IL-6, and I will come back to this in a second. Okay, then there's one thing which is also rather new and which you might have heard or not, the cleavage site, where ADAM17, the protease, cleaves the IL-6 receptor, is here between glutamine 357 and aspartic acid 358. This we determined many years ago. Now, a SNP, a single nucleotide polymorphism, has been kind of identified, which changes this aspartic acid to an alanine. And curiously, when this is done, we recently published that the cleavage of the IL-6 receptor is much more effective, meaning that you find about twice as much of this receptor whenever you have here an alanine at the cleavage site, and you found about half of this when you have an aspartic acid. So the SNP leads to higher levels, but it has been associated with a decreased risk to develop coronary heart disease or rheumatoid arthritis. There's a beautiful study, and actually this is not the only one, but I just show you this one because it's really very carefully done. Look at the number of patients they screened, and they found whenever you have this alanine at the cleavage site, you have a very much decreased risk, for instance, to develop rheumatoid arthritis, but also other diseases. So how might this work? I just told you the soluble receptor actually could make this trans-signaling response, but here it seems to be protective. So we think that it works as follows. So again, here you have the cleavage site, and an increase in the soluble IL-6 receptor concentration by enhanced shedding has actually two possible consequences. Consequence one is the cells which express the IL-6 receptor, they are less responsive because the receptor is cleaved. It's now not there anymore. But the second, which we believe is more important, there is an increased capacity of the IL-6 buffer in the blood, meaning that IL-6 can be bound to more soluble receptor, can be neutralized by the natural soluble GP130, and therefore the threshold for IL-6 to do something is much higher. But anyway, these data clearly show, which I just showed you, clearly show that the soluble IL-6 receptor levels are actually important for the susceptibility to inflammatory diseases. Now let's come back to the buffer very shortly. So when you take into account the molecular weight of the proteins, you have here IL-6, which is 20 kilodalton, the soluble receptor is 55 kilodalton, the soluble GP130 is 100 kilodalton. Now from the amounts which I showed you, you can say this is, if you set this as 1, then you have a 3.5 excess of soluble GP130. But when you take into account that you actually have a dimer of this, you have to say that it's only 1.6-fold excess of soluble GP130 versus soluble receptor. And IL-6 could now increase up to this amount and still be trapped by this, and then trapped by this, and then it will be neutralized. But you know that during inflammatory states, especially during septic states, IL-6 can actually go up to microgram levels. So it can go up 1,000,000-fold. This is actually very unusual. I think there's not a single mediator in the human body which can increase that much in cases of emergency. So this might be important. But also a question might be asked and said, why then, if this is true, what I just told you about the buffer in the blood, why then does soluble GP130 inhibit inflammation to begin with? And the reason for this, we believe, is that in every single instance where we have looked, the soluble receptor is somehow increased during inflammatory states. So it increases by the factor of 2 to 5, sometimes 10, but in every single autoimmune or inflammatory situation, the soluble receptor is increased, and then it kind of accesses the level of the natural soluble GP130, which normally stays about the same, and therefore, when you apply more soluble GP130, as you will see in a minute, this kind of blocks inflammatory states. So, now, what is now the role of IL-6 signaling in inflammatory diseases? So what we did was a very simple-minded approach. We are very much interested in IL-6, so we said we should take animal models of human inflammatory diseases such as arthritis, atherosclerosis, inflammatory bowel disease, many others, and first of all perform these models in mice which have no IL-6. They are called IL-6 knockout mice, so the gene has been destroyed, or we neutralize IL-6 with an antibody against IL-6 or the IL-6 receptor. And we asked whether when we do this, whether the disease still occurs. And then, second approach, in parallel, we just do the very same, but now we do this with a soluble GP130 Fc protein, which I just told you, which especially or specifically only inhibits trans signaling, or we use mice which we generated, which are transgenic for this protein, and therefore they have no trans signaling. And again, we ask, okay, what about the model we are looking at? And I'll show you one of these examples, a very recent example, which has just been published a couple of weeks ago. It's a nephrotoxic nephritis model. And what you do is actually you inject mice with an anti-serum which has been raised against podocytes, which is a cell population in the kidney. And when you do this, and you do this in the presence of anti-IL6 or soluble GP130, you see that here the situation seems to be much worse, whereas here the kidney looks very nice. And this can actually be quantified. You can quantify the renal damage, and you can quantify the renal function. And it seems when you have no IL-6, you actually have less renal function and more renal damage. So in this case, IL-6 seems to have a regenerative effect. Very, very interesting. And when you look at this a little bit more closely, you can ask which kind of cells are actually those which express the IL-6 receptor. And very clearly, these are macrophages, kind of inflammatory macrophages characterized by this marker here. And indeed, when you look what happens when you treat these cells with IL-6, you see that you decrease with IL-6 the proliferation of these inflammatory macrophages. And when you look at these macrophages from mice which have no IL-6, from IL-6 knockout mice, you see that you have then more of these inflammatory macrophages, somehow confirming what I showed you in the last slide. IL-6 seems to be beneficial in this kind of animal model. And the final point was when we just knock out the IL-6 receptor only on macrophages, and then again compare the renal damage of this kind of model, you clearly see when you have no IL-6 receptor on these cells, then you have much less damage and you have more renal function. And actually this has to be seen in the context of that IL-6 stimulation of these macrophages via, importantly, the membrane-bound IL-6 receptor executes an anti-inflammatory activity which goes via the kind of differentiation between M1 and M2 macrophages. You know, M1 are very inflammatory, M2 are rather regenerative, and this seems to be under the control of IL-6, which means that not always blocking IL-6 is important. And when we did this with really many animal models, and you can see they really range from inflammatory bowel disease through rheumatoid arthritis and through several cancer models and also even an obesity model. I will show you this in a minute, and the nephrotoxic nephritis model I showed you in a couple of minutes ago. So in all of these models, it turned out that only the blockade of trans signaling was enough to completely block the maintenance or the progression of the disease. And this obviously led us to a model where we said that the activity of IL-6 when it goes via the soluble receptor, this seems to be a pro-inflammatory activity, whereas the activity of IL-6 when it goes via the membrane-bound receptor, this seems to be rather regenerative or protective or anti-inflammatory. And this, if true, would already argue that it might be much better just to block this part here than to globally block IL-6. And I will come back to this in a second. So you know, or you should know, that blocking IL6 in patients actually has two problems. And one problem is shown here, or mentioned here. These are bacterial infections. And this is just one example. So during this particular trial, more than 20% of the patients had problems with bacterial infections. So therefore, we did exactly the same kind of experiment which I showed you 5 minutes ago. So we infected mice with bacteria. We blocked globally IL6 with anti-IL6 antibody, and we just blocked trans-signaling with soluble GP130. And the positive control is actually shown here. This is the expression of one of these liver acute phase proteins. It's called serum amyloid A. It's like in the human CRP, so it shoots up when something happens in the body. And you can see the expression of this is blocked when you neutralize globally IL6. It is not affected when you just block trans-signaling, and the reason for this is that hepatocytes do express, as I showed you in the beginning, do express the IL6 receptor. So therefore, this protein does not block. But we are not interested in this protein. We are interested in the progression of the infection control, and you can see here, when you now look at the number of bacteria you find in spleen and liver, they are here, this log scale, they are about 1 log higher when you globally block IL6, and they are not affected at all when you just block trans-signaling. So this seems to argue that the protection from bacterial infection seems to rely rather on classic signaling, not on trans-signaling, and this is something you do not want to block. And the last example I show you is something which is very new data, very, very intriguing data. You probably heard, or you have the experience, when you block IL6 in patients very often gain weight, and cholesterol goes up and triglycerides go up. And therefore, we also kind of addressed this kind of situation. So we had in mind a very beautiful experiment which had been done by a German group, not by us, Matthias Blüher from Leipzig in Germany. And what he did is, you all know that when people have a very high BMI, we are talking between 40 and 50. This is really a lot. It's about, about this size. So when you have people who have such a high BMI, many of these are insulin resistant, but not all of them. So this study now compared what happens, or what is actually the difference if you have such a high BMI and what makes you insulin resistant or sensitive. And what they found is actually that the shape of the fat is different. So this person is insulin sensitive, this one is insulin resistant. So this is much more flexible than in this case. But the main difference is actually when you look at the adipocytes, and what you see here is that in this case, in the sensitive case, the adipocytes have not these structures which you see here, and these are actually infiltrating macrophages. So infiltrating macrophages in an obese person seem to make a sort of chronic inflammatory state, and this seems to have to do very much with insulin resistance. So when you look at people who are lean, at people who have an impaired glucose tolerance, and at people who are type 2 diabetic, and when you look at the soluble receptor, this goes up very much and very significantly. And you can actually make a correlation, and you can also look at the HOMA, which is also a measure for the kind of insulin resistance or sensitivity, and you clearly see that you get a straight correlation between HOMA and plasma soluble IL6 receptor levels. Very interesting. And therefore, we made again an animal experiment in the way you saw it before. So these are normal mice, and they are fed a normal diet, and then they are fed a high-fat diet. And when they are fed a high-fat diet, they develop exactly what you saw in this person who was insulin resistant. You get these macrophages, and the metabolic people call this a crown-like structure. And then we do the very same experiment in mice which are either treated with soluble GP130 Fc or which are transgenic for the same protein, just asking whether blockade of transsignaling has to do with this. And you clearly see there are no crown-like structures at all. When you quantify it, it's very, very highly significant and it might be very important. So why do we think this is important? We think that IL6 in the liver has a very important metabolic role. So we know, and this actually very fits to the patient data, that IL6 minus mice which have no IL6 gene, they get obese later in life. We also know that IL6 minus mice show glucose intolerance, insulin resistance, and liver inflammation. We also know that IL6 ablation only, IL6 receptor ablation only in the liver leads actually to more or less the very same phenotype. Again, glucose intolerance, insulin resistance, and liver inflammation. And we also know that IL6 gene transfer, so if you have more IL6 signaling or more IL6 in the body, that reverses body weight gain and fatty liver in obese mice. And liver toxicity also has been reported upon treatment of patients with this already mentioned tocilizumab. So there seems to be a very, very strong connection between liver, IL6, and metabolism. And therefore, we were very pleased actually to see that in our case, the selective blockade of IL6 trans-signaling did not exacerbate high-fat diet-induced increases in circulating non-esterified fatty acid concentration, did not increase in hepatosteatosis, liver inflammation, or liver function. This might be very important because this is really something, I mean, in very general terms, you all know, when you give a drug or you give a biologic to a person with an autoimmune disease, you don't give it once, you give it probably for 25 years. So bacterial infections and metabolic effects might be really important issues, and we think these are very important points. So what I showed you so far is, I said that trans-signaling via the soluble receptor is specifically blocked by this protein which we made, that classic but not trans-signaling is protective in bacterial infections, that blockade of IL6 trans-signaling prevents infiltration of macrophages into adipose tissue, and therefore we would like to hypothesize that this protein will be the next generation drug for the treatment of, for instance, inflammatory and autoimmune conditions. And we not only hypothesized, we also did something. So we founded a small company in Kiel, which is called Conaris, and this Conaris made a license agreement with a bigger company in Copenhagen, Ferring Pharmaceuticals. And now we are kind of on the way, so it took us some time. It was actually very, very difficult to make a GMP production. It was also very expensive, and Ferring paid actually for all of this. Then we went through the non-clinical safety trials. We already have finished the phase 1 trials in 2013, 2014. So there has been no kind of adverse effect, so everything went fine, and we have a very good half-life of the compound. And now in 2015, we will start with phase 2 of this protein, which specifically blocks the IL6 trans-signaling. And now, since we are closely watching the IL6 market, it turns out that now many people, or many companies, and some of them are now kind of represented outside in the exhibition, many companies now develop tools to block IL6, but what you should see is all of these being anti-IL6 or anti-IL6 receptor monoclonal antibodies, they all block both classic and trans signaling. So if that is true, what I just told you, it is probably much better or much more beneficial just to block this IL6 trans signaling with a protein which we made, which has now a different name, FE something. And this is actually also shown in the review which I mentioned in the beginning. So all of this work has been funded. It was actually very expensive by mostly German or European agencies. So these are the networks where we are kind of represented in. Then we had a lot or I had a lot of help of my co-workers, and I just want to mention a few. So Athena and Christoph, they actually made most of the work together with these two graduate students, Jeanette and Stephanie. Björn Rabe, he actually a couple of years ago generated these mice which are transgenic for the soluble GP130 protein. And Jürgen and Thomas, they already left the lab, but they were very important in the generation of the first generation of the soluble GP130 protein. And we had a lot of external help by Jürgen Scheller, who was actually in my lab, is now director of biochemistry in Düsseldorf, also in Germany. And these people did the toxic nephritis model, and these helped with the bacterial infection, and these helped with the metabolic studies. And with this, I would like to thank you very much for your attention. This is actually where I live in Germany, beautiful. Again, summer better than winter, so please come visit in summer, and I'm happy to answer your questions. Thank you very much.